Very few phenotypes are the result of the action of just a single gene. Most phenotypes are in fact produced by multiple genes acting together, and not only do these genes act together, they also interact with each other. So let us start by looking at interactions between different alleles of the same gene. But before we start discussing gene interactions, let's look at plain old recessiveness. We understand that the recessive phenotype is only apparent or only shows in the individual who's homozygous for the recessive allele. But what is the molecular basis of recessiveness? And we can understand this with the example of a disease called phenylketonuria or PKU. In normal or wild-type individuals, from dietary protein, phenylalanine is extracted. This phenylalanine is converted into tyrosine by an enzyme called phenylalanine hydroxylase. And just to remind you, an enzyme is nothing but a protein that can catalyze biochemical reactions, which means it can make biochemical reactions go faster. And the job of phenylalanine hydroxylase is to convert phenylalanine into tyrosine. Now, phenylalanine hydroxylase is encoded by this gene called PAH, and the wild-type alleles, the big P alleles in humans, they encode a functional enzyme that can then convert phenylalanine into tyrosine. However, if you have the mutant allele, little p, of the PAH gene, then you do not make PAH. So these individuals, the mutant individuals, have no PAH, and therefore there is a block to this conversion of phenylalanine into tyrosine that leads to a buildup of phenylalanine, which gets converted to phenylpyruvic acid, and that accumulates in the brain and causes developmental delays and ultimately mental retardation. And this condition is known as PKU. So, what happens in the heterozygous individual? Since the big P allele makes the enzyme, whereas the little p allele does not make the enzyme, these individuals will have half the amount of phenylalanine hydroxylase, half the amount of the enzyme. In other words, they will have one dose. And this one dose is sufficient to convert phenylalanine into tyrosine. So even though you have half the amount of the enzyme, this amount happens to be sufficient for converting the phenylalanine into tyrosine and avoiding a buildup of phenylalanine and conversion to phenylpyruvic acid, leading to mental retardation. And that is why the big P allele, the one that makes the enzyme, is dominant, whereas the little p allele that does not make the enzyme is recessive. And this way of having recessiveness is called haplosufficiency, and haplo means one dose. And so the heterozygous individual has just one dose of the functional gene that makes the enzyme. However, that one dose happens to be sufficient for having a wild type phenotype and not having the disease. Having looked at how recessiveness arises at the molecular level, let's discuss how dominance can arise at the molecular level. Now, there are two ways of having a dominance at the molecular level. One is the opposite of haplosufficiency, that means haploinsufficiency. And there is a second mechanism, dominant negative interactions, that can give rise to dominance. First, let's discuss haploinsufficiency. And as we said, it's the opposite of haplosufficiency. So let's say that one dose in the het was not sufficient to convert phenylalanine into tyrosine, and you really needed the full amount produced by the two big P alleles to successfully convert phenylalanine into tyrosine, then these heterozygous individuals would have had a buildup of phenylalanine, which would have gotten converted to phenylpyruvic acid, and which would have accumulated in the brain, giving these individuals PKU. And then the little p allele would have been dominant rather than being recessive, since the het would be affected by PKU. And so that's haploinsufficiency, where one dose is not sufficient for achieving the wild type phenotype. Now, the other kind of dominance that occurs by dominant negative interactions between alleles can be understood with the example of collagen. So the normal wild type allele, and I'm using little c to denote that wild type allele, encodes this collagen protein that makes trimers. That means three protein molecules, they bind together, they attach to each other in order to make a complex that is functional. However, if you have the mutant allele, which I'm denoting with big C, this mutant allele encodes a protein that binds to two of the wild type proteins and prevents these proteins from forming a trimer. So in the wild type, collagen makes a trimer, but when you have this mutant allele, big C, it prevents the trimer from forming, leading to the mutant phenotype. And therefore this allele big C affects the wild type, the function of the wild type protein, in a negative manner, and causes a dominant phenotype, because you only need one copy of the big C allele in order to interfere with the normal trimer formation of collagen and to give you a mutant phenotype. And this is the second mechanism by which you can get complete dominance. The next type of interactions between alleles we have seen previously in our discussion of polygenic traits, and that is incomplete dominance. And in incomplete dominance, the heterozygous genotype has a phenotype that is intermediate to the phenotypes of the two alleles. To go back to our example of the color of wheat kernels, let's say big R1 homozygotes have red kernel color, whereas little r1 homozygotes have white kernel color or no pigmentation, then the hets big R1 over little r1 will have light red or pink color. And this can be understood once again in terms of doses, so that the individual who's homozygous for the big R1 allele has 2 doses makes more pigment than the individual who has only 1 pigment making allele, the heterozygous individual, who makes more pigment than the individual who is homozygous for the allele that does not make any pigment. The next type of interaction between the alleles of the same gene is something called codominance. Now, the best example of codominance is the ABO gene and human blood types. There are 4 major blood types: A, B, O, and AB. And these blood types are determined by these antigen proteins that sit across the plasma membrane of our red blood cells. To these antigen proteins are attached different sugars, and the ABO gene determines which sugar will be attached, or the allele of the ABO gene determines which sugar is attached to the antigen protein. Now, when someone donates blood and the recipient has an incompatible blood type, then antibodies in their serum will recognize these sugars on the donor's red blood cells and cause agglutination or clumping up and causing incompatibility of the 2 blood cell types. So the first interesting thing about the ABO gene and its alleles is that we don't just have 2 alleles, but in fact there are 3 alleles: IA, IB, and little I. And having more than 2 alleles has a name, and it's called multiple allelism. Now, if you are IA over IA, then of course you have the blood type of your blood group of A, and IA is dominant over little I, so that the het also has the blood type or blood group A. If you are IB over IB, and there's a little typo here, this should be a B. If you're IB over IB, then you are blood group B, and IB is also dominant over a little I, so that the het has blood group B. Little I is a recessive allele, so if you're homozygous for little I, then you have blood group O. And the interesting thing that happens is that if you happen to be a het between IA and IB, then you have both the sugars attached to your antigen proteins on the surface of red blood cells, and therefore you show the phenotype of both the IA allele and the IB allele, and this is known as codominance, which gives you an entirely different blood group, AB, that's different from the blood groups of each individual allele. Let's practice with the problem. In a maternity ward, 4 babies become accidentally mixed up. The ABO types of the 4 babies are known to be O, A, B, and AB. The ABO types of the 4 sets of parents are determined indicate which baby belongs to each set of parents, and we are given 4 different parental crosses here. And so the first step here would be to actually write down the genotypes of the parents and hence the cross. So let's say I have AB crossed with O. That means AB blood group is produced by individuals who are heterozygous for the IA and IB alleles, whereas the O blood group is produced by individuals who are little I over little I, and that means the progeny of this cross, you can have 2 types of individuals, IA over I or IB over I, and IA over I will be blood group A and IB over I will be blood group B. Now we can go and work out the next cross. We have A over O, and that means our cross is IA over IA or IA over I cross to little I over little I, and in this case we have 2 types of progeny that are possible. We have IA over I or little I over little I, depending on whether the A parent is IA over IA homozygote or IA over little I het. And so we could get either the A or the O. Then in the third case, we have A cross to AB, and that implies that we are crossing an IA over IA individual or IA over I to an IA over IB individual, and we have in this case a few different genotypes that are possible. You could get IA over IA with an A blood group. You could get IA over IB with an AB blood group, and you could get an IB over little I individual, and these 3 are all the genotypes that could result from this cross. And finally, we have the O cross to O individual, and in this case we are crossing 2 little I homozygotes, and since they're both homozygotes, only one type of progeny is possible, that is little I over little I or the O blood group. Now each set of parents has exactly 1 child, so if this set of parents, the 2 parents who are blood group O, can only have an O child, it means that this A individual who was crossed to O cannot have an O child and their child must have blood group A. And that allows us to eliminate blood group A from the first cross, telling us that the child with the blood group B is from the parents, one having the blood group AB and the other having the blood group O, and this eliminates both A and B from the third cross, telling us that the parents who are blood group A and blood group AB have the child who has the blood group AB.